For centuries, humankind has carried a quiet curiosity: why do some illnesses seem to run in families? Even Hippocrates, over 2,000 years ago, suspected that the answer might lie within us. As time passed, the mystery began to unfold. Researchers discovered that the secrets of inheritance were written in our genes, tiny scripts inside the vast book of life. Now, that book is being read in an entirely new way. Scientists at EMBL, alongside their collaborators, have unveiled a powerful tool that pushes single-cell technology further than ever before. This tool doesn't just read genetic code, it listens to the whispers of DNA and RNA within the same single cell. For the first time, researchers can see both the blueprint and the message it sends, side by side. By focusing on the non-coding regions of DNA, the parts once dismissed as junk, the tool reveals where disease-linked variations most often hide. These silent regions, long overlooked, are turning out to be the key to understanding countless illnesses. This has been a long-standing challenge, says Dominic Lindenhofer, lead author of the study published in Nature Methods. Earlier single-cell tools could only detect variants from expressed genes. Our method captures them all, even those buried deep in the genome. To understand why this matters, think of the genome as a grand library. Some books, the coding regions, tell the cell exactly how to make proteins, the building blocks of life. But most of the library, the non-coding sections, contains instructions on when and where to use those books. And here's the striking truth: more than 95% of disease-related variants occur in those non-coding regions. Yet until now, scientists couldn't study DNA and RNA together from the same cell to uncover their true effects. In this non-coding space, Lindenhofer explains, lie variants linked to conditions like autism, schizophrenia, and congenital heart disease, still vastly unexplored. We needed a tool to uncover their stories. That tool is SDR-seq, a breakthrough that merges DNA and RNA sequencing at the single-cell level. It works by trapping each cell inside a tiny droplet of oil and water, a miniature reaction chamber where both DNA and RNA can be read at once. Behind this innovation lies a symphony of collaboration. Scientists from EMBL's Genome Biology and Computational Biology units joined forces with researchers from Stanford University and Heidelberg University Hospital. One team learned how to fix cells to protect fragile RNA. Another built a complex decoder to interpret the intricate DNA barcodes, digital fingerprints of each single cell. And although this decoder was designed for this project, it could soon empower many others in genomics research. To test the tool's power, the team turned to B-cell lymphoma, a type of blood cancer rich in genetic variation. By observing thousands of individual cells, they discovered something remarkable: cells with more genetic mutations had stronger growth signals, pushing them toward a more malignant state. We can now see whether a variant exists on one or both copies of a gene, says Lindenhofer. And we can directly link it to how that gene behaves inside the same single cell. This insight changes everything. For the first time, scientists can connect the cause, a tiny change in DNA, with the effect, how that change drives disease. As Lars Steinmetz, senior author and EMBL group leader, explains, we now have a tool that can link variants to disease. If we understand how these variants regulate disease, we can finally learn how to treat it. The impact of SDR-seq is only beginning to unfold. From decoding genetic mysteries to designing precise diagnostics, this innovation marks a new era in genomics where every cell tells its own story, and science listens closer than ever before. And so, with SDR-seq, humanity takes one step closer to reading the hidden language of life itself, unlocking the codes that shape our health, our future, and the very essence of who we are.